Dolly the sheep, Scotland, 1996. At the Roslin Institute near Edinburgh, a lamb was born that would change biology forever. Her name was Dolly. She looked like any other sheep, but she wasn't born the normal way. Dolly was a clone, the first mammal ever created from a single adult cell. The process was called somatic cell nuclear transfer. Scientists took a cell from the mammary gland of an adult ewe, removed its nucleus, and placed it inside an empty egg. That egg grew into a lamb that was a perfect genetic copy of the sheep the cell came from. No father, no mixing of DNA, just one cell rebuilt into a whole animal. It almost didn't work. It took 277 attempts. Dolly was the only success. When she was revealed to the world in 1997, she caused a global storm. Suddenly, cloning wasn't science fiction. People asked the obvious question: if you can clone a sheep, can you clone a human? Dolly lived 6 years. She had lambs of her own, and when she died, she was preserved in a museum where she still stands today. One sheep that proved life could be copied. The Soviet silver fox experiment. Siberia, 1959. A Soviet scientist named Dmitry Belyayev began one of the longest experiments in the history of biology. He wanted to answer a simple question: how did wild wolves become friendly dogs? So he started with silver foxes, wild, aggressive, and terrified of humans. Every generation he did one thing. He picked only the calmest foxes and let only those breed. Nothing else. No genetic engineering, just selecting for friendliness. And something incredible happened. Within just a few generations, the foxes started to change, not only in behavior, but in their bodies. Their ears became floppy, their tails began to curl and wag, their fur developed spots and patches. They started barking and seeking out human attention. They were turning into dogs right before the scientists' eyes. By selecting for a single trait, tameness, Belyayev had accidentally recreated the entire process of domestication. The experiment proved that behavior and physical traits are deeply linked in DNA. Belyayev died in 1985, but the experiment never stopped. Those foxes are still being bred today, some so tame you could keep one as a pet. Spider goats. Spider silk is one of the strongest materials in the natural world. Pound for pound, it's tougher than steel and more flexible than rubber. The problem? You can't farm spiders. Put too many together and they eat each other. So scientists found another way. At a company in Canada, researchers took a single gene, the one spiders use to produce silk, and inserted it into goats. The result was a herd of animals that looked completely normal, but produced spider silk protein in their milk. They would milk the goats, extract the silk protein, and spin it into fibers they called biosteel. The goal was extraordinary: material for bulletproof vests, artificial tendons, medical sutures, even parachute cords. The goats themselves were healthy. They had no idea they were carrying a piece of a spider's genetic code. The company eventually went bankrupt, but the research survived, moving to universities that continue it to this day. It remains one of the strangest hybrids ever created. Part farm animal, part spider, walking around a field like nothing happened. The glowing animals. In the ocean lives a jellyfish that produces a glowing green protein. Scientists discovered they could take the gene for that glow and put it into almost anything. They started with mice. By inserting the jellyfish gene, they created rodents that glowed bright green under ultraviolet light. It wasn't just for show. The glow acted like a marker, letting scientists see exactly which genes were switching on or off inside a living body. Then it spread. Glowing fish, glowing rabbits, glowing pigs, cats, and even monkeys. One rabbit, named Alba, was created as a piece of art, a living animal that glowed green in the dark. And the fish became famous too. Originally designed to detect pollution in water, they were turned into pets, the glowfish, becoming the first genetically modified animal ever sold to the public. Today, you can buy them in a pet store. What started as a jellyfish floating in the sea became a tool that transformed medicine and filled aquariums around the world with animals that shouldn't be able to glow, but do. The earmouse. In 1997, a single photograph horrified the world. It showed a laboratory mouse with what looked like a full human ear growing out of its back. People assumed the worst, that scientists had fused a human and an animal. The truth was stranger and less sinister. There was no human tissue and no genetic fusion. Researchers had built a tiny ear-shaped scaffold out of biodegradable material, seeded it with cartilage cells, and implanted it under the skin of the mouse. The cells grew, filling the mold until it formed a perfect ear shape. The mouse used was special, bred to have no immune system, so its body wouldn't reject the implant. The purpose was regenerative medicine. Imagine growing a new ear for someone born without one, or a new nose, or replacement cartilage, built from a patient's own cells. The ear mouse was proof that human body parts could be grown outside the human body. It became one of the most famous and most misunderstood science images ever taken. A mouse that looked like a monster, but was actually a glimpse of the future of medicine. Super-muscular dogs! Inside every animal is a gene called myostatin. Its job is to put a limit on how much muscle the body can grow. But what happens if you switch that limit off? In 2015, scientists in China decided to find out. Using gene editing technology called CRISPR, they disabled the myostatin gene in a litter of beagles. The result was a new kind of dog, unusually strong, with dramatically more muscle than a normal beagle. They called one of them Hercules. With their limiter removed, the dog's bodies just kept building muscle, making them far more powerful than nature intended. Officially, the goal was medical, creating models to study human muscle diseases like muscular dystrophy. But the researchers admitted these dogs could have other uses too. Stronger police dogs, military dogs, working animals. It was one of the first times gene editing was used to physically redesign a common pet. And it raised a question that still hangs over the field. If we can edit a dog to be stronger, what exactly are we willing to edit next? AquAdvantage salmon. A normal Atlantic salmon takes about 3 years to reach full size. Scientists thought that was too slow, so they engineered a fish to beat nature. They took a growth hormone gene from a Chinook salmon and a genetic switch from another fish called the ocean pout. Together, these kept the salmon's growth hormone running all year long, instead of only part of the time. The result? A salmon that grows to market size in about half the time. Critics gave it a nickname, Frankenfish. For decades, it was blocked, debated, and feared. But eventually, it made history. The AquAdvantage salmon became the first genetically engineered animal ever approved for people to eat. To ease fears of it escaping and breeding with wild fish, the salmon are raised in enclosed tanks on land and engineered to be sterile. You may have already seen GM crops on your plate, but this was different. For the first time, an animal itself had been rewritten to grow faster, cheaper, and land on your dinner table. The Enviropig. Pig farming has a hidden problem. Pigs can't fully digest the phosphorus in their food, so it passes through them and into their manure. And when that manure runs into rivers and lakes, the phosphorus fuels massive toxic algae blooms that suffocate everything living in the water. In Canada, scientists thought they could fix this at the source, inside the pig itself. They engineered a new kind of pig that produced a special enzyme in its saliva, letting it break down phosphorus before it was ever wasted. They called it the Enviropig. The results were impressive. Its manure contained up to 60% less phosphorus. It was a genetically modified animal designed not for food or profit, but to protect the environment. But it never made it to market. Approval was slow, funding dried up, and public fear of Frankenswine never faded. In 2012, the project was shut down and the last Enviropigs were put down. Only their genetic material was frozen and saved. It remains one of the great what-ifs of genetic engineering, an animal built to heal the planet that the world simply refused to accept. The cloned monkeys. Cloning a sheep was one thing. Cloning a primate, an animal much closer to us, was far harder. For over 20 years, scientists tried and failed. Then, in 2018, a lab in China succeeded. They produced 2 identical monkeys named Zhang and Hua using the exact same technique that created Dolly the sheep. They were the first primates ever cloned from a body cell, and it didn't stop at cloning. A few years earlier, scientists had used gene editing to to create the first monkeys with deliberately altered DNA, engineered to carry specific genetic mutations. The purpose was medical. Monkeys are biologically similar to humans, so genetically identical edited primates could become powerful tools for studying diseases like Alzheimer's, autism, and cancer. But the implications were impossible to ignore. These were animals one step away from us on the evolutionary tree. If you can clone a monkey and edit its genes at will, the line between scientific breakthrough and something far more unsettling starts to disappear. And that line is exactly where this research now sits. The oncomouse. In the 1980s, scientists at Harvard created a mouse unlike any before it. They engineered it by inserting a cancer-causing gene directly into its DNA, so that the mouse and all its offspring would be highly likely to develop tumors. They called it the oncomouse. It sounds cruel, and in a way it was, but it was also one of the most valuable tools in the history of medicine. Because this mouse reliably developed cancer, researchers could finally test new treatments and drugs on a living model that mirrored human disease. Countless cancer studies depended on it. But the oncomouse made history for another reason. In 1988, it became the first animal ever granted a patent. For the first time, a living, breathing creature could be legally owned as an invention. That decision started a global debate that still isn't settled. Can you patent life? Should a company be allowed to own an entire species it designed in a lab? The oncomouse was more than an experiment. It was the moment genetically engineered animals became property. GM mosquitoes. Mosquitoes are the deadliest animals on Earth, not because of what they are, but because of what they carry. Dengue, Zika, yellow fever, diseases that kill hundreds of thousands of people every year. For decades, we fought them with poison. Then scientists tried something new. They didn't try to kill mosquitoes, they rewrote them. A company engineered male mosquitoes with a hidden genetic flaw, a self-limiting gene. These modified males were released into the wild by the millions. When they mated with wild females, they passed on the gene, and their female offspring died before they could grow up and bite. Fewer females meant fewer mosquitoes, and over time, local populations began to collapse. It was tested in Brazil, the Caribbean, and even Florida. In some areas, mosquito numbers dropped by more than 90%. But releasing a genetically modified animal into the open world, on purpose, terrified a lot of people. Once those genes are out there, you can't take them back. It's one of the boldest experiments ever attempted, using genetic engineering not in a lab, but on the entire planet. The woolly mammoth. Every animal on this list was already alive. This one has been extinct for thousands of years. A company in the United States has set out to do what sounds impossible: bring back the woolly mammoth. They can't clone it, there's no living mammoth to copy from. So instead, they're rewriting its closest living relative, the Asian elephant. By editing the elephant's DNA with mammoth genes for thick fur, cold-resistant fat, and small ears, they hope to create a living, breathing, mammoth-like animal. And in 2025, they took a first step that stunned the world. They engineered mice with mammoth-inspired traits, covered in thick, woolly hair. The woolly mouse was proof that the genetics could actually work. The full mammoth is still years away, and they don't plan to stop there. The dodo, the dire wolf, and other lost species are all on their list. It's the ultimate genetic experiment, not editing life, not copying life, but resurrecting it. For the first time in history, extinction might not be the end.